Welcome back, guys. So, just now we have mentioned about the physiology of the synthesis and secretion of the thyroid hormones. So, before we move on into the action of the thyroid hormones, I just want to mention about how the thyroid hormones are actually transported once they are actually in the circulation. Okay, okay. The 70% of the thyroid hormone that is released, they are actually bound to thyroid binding globulin. This is TBG, which is different from thyroglobulin, TGB. Okay, 70% of the thyroid hormone, no matter T4 or T3, they are actually bound to thyroid binding globulin. Okay, so another 10 to 15% is actually transported by another hormone called as the transthyretin, and the remaining 15% actually can be carried by the albumin. So T4, okay, one important thing that you have to remember is T4 binds more tightly than T3. So what is the importance of this? So although T3 is more biologically active, it has a very short half-life. So roughly it is around 1 day. So T4 has a longer half-life, which is around 7 days. So they can buffer any acute changes in the thyroid gland function. For example, when your thyroid gland suddenly shuts down. So as a summary of what I've talked about, so the lesser the or the lower the extent of the binding protein levels, the higher the free thyroid hormone is in the circulation. Therefore, the release of the thyroid stimulating hormone would be lower, and the thyroid hormone synthesis and release as also would be lower. Okay, so and okay, now we can go on to the action of the thyroid hormone. Okay, this picture is just showing a summary of the action of the thyroid hormone from the circulation into the cells, into the cells, target cell, for example liver, pituitary, brain, as well as the heart. Okay, so when the hormones were in the circulation, they are bound either to thyroid binding globulin or transthyretin, or even can be with albumin. Okay, once T4 and T3 leave the plasma, they enter the cell either by diffusing through the lipid of the cell membrane or by carrier mediated transport. So they actually can actually diffuse across this cell membrane, okay, by just normal diffusion, or they can also get diffused into by the carrier mediated transport. Once T4 is inside the cell, we know the active form of the thyroid hormone is T3. So the T4 need to be converted to T3. So inside the cytoplasm, we have an enzyme called the 5'-3' monodeiodinase enzyme, which converts the T4 to T3. As you can see here also we have an enzyme called deiodinase, T4 to T3. Here the name of the enzyme is actually quite different, but the function is just the same. The name of the enzyme is 5'-3' monodeiodinase, which converts T4 to T3 by removing. Why we call it as 5'-3' monodeiodinase is because they remove the 5' iodine through deiodination. So now the T3 hormone is actually inside the cytoplasm. Okay, this is actually the nucleus. The nucleus actually can have TR genes or thyroid genes in the DNA. So different places have different genes. For example, in liver we have TR gene alpha, and in brain, in liver we have TR gene alpha, and in brain we have TR gene beta. Okay, and as you can see here, there is a receptor that is bound to the nucleus. So the receptor that binds to DNA, okay, this is the DNA. The receptor, this whole receptor that is bound to the DNA is preferentially a heterodimer of a thyroid hormone receptor and retinoid extract receptor, as you can actually see here. Or to be specifically, it is T3 receptor, because only T3 is biologically active. Okay, so upon the binding, okay, now we have here T3. T3 actually diffuses through the nuclear membrane and gets bound to the T3 receptor. So upon the binding of T3 hormone onto the ligand binding domain of the thyroid hormone receptor, the compressor at the thyroid receptor is kicked out. Okay, there is something called a compressor here. Let me just okay, actually there would be something called compressor here. This compressor would actually be kicked out, and there is another thing called as the RNA polymerase. Okay, let's say the RNA polymerase is inside the nucleus also. Okay, let's say this RNA polymerase, and as well the co-activator. Okay, RNA plus co-activator. I'll just put co-A. Okay, this one is the compressor. Once T3 bound to the T3 receptor, compressor will actually get kicked out, and RNA plus co-activator will actually bound to this T3 receptor. So now this RNA and co-activator is associated with this heterodimer of the retinoid X receptor and T3 receptor. So when this thing has been, when all this thing is present, then only transcription of this area would actually occur, and this transcription produce mRNA, which is translated to produce proteins. Okay, what are those proteins? These are the proteins that usually would be formed. That is glycerol 3-phosphate dehydrogenase, which, if you remember, if you did your biochemistry, you know that this enzyme is actually present in the glycolysis pathway. Okay, this is the first enzyme that is produced, and another enzyme that is produced is actually the Na plus K plus ATPase. Okay, there is actually, let me just change this first. There's actually a few function of why these enzymes are actually being formed. Okay, these two enzymes play important role in the metabolism of a few things such as carbohydrate, protein, lipid, as well as this Na plus K plus pump itself. So as for the carbohydrate metabolism, the thyroid hormone actually raises the rate of hepatic glucose production by increasing the gluconeogenesis. And as for the protein metabolism, the amino acids required for the gluconeogenesis comes from the increased proteolysis in the muscle. So this also increases the protein synthesis, but this is outweighed by the proteolysis. Okay, and that is for protein metabolism. And the next one is the lipid metabolism. Okay, thyroid hormone increases the degradation of the stored triacylglycerols or triglycerides in adipose tissue, releasing fatty acids as well as glycerol. This fatty acid provide fuel for liver to support gluconeogenesis, and the glycerol, whereas the glycerol provides the starting material for the gluconeogenesis. So in essence, by promoting all these three processes, they stimulate energy and O2 consumption, which is why you can see those people who is actually having hyperthyroidism, they usually would be having bigger appetite, but they would not really increase in size, which is why you guys might get jealous, because you guys probably will just eat just a few stuff maybe, and you're already looking at like a very big in size or something else. So okay, that is an advantage of being a hyperthyroid patient. No matter how much you eat, you're just going to be thin. So okay, back to this the last effect of the new enzymes that is produced by this thyroid hormone is, what is its effect on the Na plus K plus pump. So it increases in the O2 consumption, because the NKA activity is actually increased, therefore the O2 consumption would eventually increase. Okay, so these are the other functions that I've talked about. Some of this I have also talked about, such as it also increases the beta adrenergic receptor, which makes the heart to be more sensitive to the circulating catecholamine and eventually increasing the heart rate. And oxygen consumption, as I've talked before, basal metabolic rate. So basal metabolic rate is very high, so that's why those people are usually thin. Hypothyroidism patients are usually thin. And temperature homeostasis, they usually their body is actually very warm, and they also would have an increased growth, okay, because there is an increased growth hormone transcription and induction of the anabolic enzymes. So those are the effect of the thyroid. Yeah, those are the effects of the thyroid hormones. So I'm just going to mention a few things else which is not in this slide. Okay, the thyroid actually produces more T4 than T3, but T3 has a greater thyroid activity and more biologically active. And the main source of circulating T3 is peripheral deiodination of T4 by deiodinases. So these deiodinase as well as these deiodinase. So 80% of the T4 are produced 80% of T4 produced are deiodinated in the periphery, which means in the circulation. Okay, uh, which means they are not actually counting this deiodinase or this iodinase. This iodinase, they are actually counting on the. They actually. Where's the pen? They're actually talking about even in the blood circulation also we have an enzyme called deiodinase. So even in the blood itself, T4 actually can be converted to T3 by the action of this deiodinase. So 80% of the T4 produced are deiodinated in the periphery. And sometimes when the T4 is actually being deiodinated, you can also produce another things also known as a reverse transcriptase. And these reverse transcriptase, basically they don't have any biological activity, so they actually would be having a higher metabolic clearance and lower serum concentration. Okay, obviously when you have a higher metabolic clearance, when you're actually removing out from your body, the amount of this thing inside your serum would be lower. So that's all for this whole two parts of the videos, which is about this synthesis, which is basically about the physiology of the thyroid hormones, how it is synthesized and secreted. I'll catch you up in other videos the next time. So thank you very much, guys.